Okay guys, I want to give you a little bit of a short video just on everything we went over in class that isn't in my other video, which is the DNA replication video. So I just want to go over pretty much the structure of DNA. And if you remember Rosalind Franklin, she was the lady who did a lot of the research that helped Watson and Crick come up with their structure of DNA as we went over in class. But we're going to go over the structure that they found. And just to begin, they started off with a 5-carbon sugar. All right, and then attached to our 5-carbon sugar is a phosphate group. And then lastly, going towards the middle, but is also attached to our 5-carbon sugar, is going to be our nitrogen base. I'm going to label all this real quick. There's our 5-carbon sugar. Now for DNA, our 5-carbon sugar is going to be deoxyribose. Okay, so for DNA, it's deoxyribose. For RNA, that 5-carbon sugar is ribose instead. All right, so that's one difference between DNA and RNA. Now, if we're looking at this, our 5-carbon sugar, it's made up of 5 carbons, obviously, but if we count them, this would be carbon 1, this would be carbon 2, this would be carbon 3, this would be carbon 4, and then up here, attached to the phosphate, that's our 5th carbon, right? And there is your phosphate. And here is your nitrogen base. I'm just going to say this is adenine. All right. After we have one nucleotide in here, so this is a nucleotide of DNA. It's made up of phosphate, a 5-carbon sugar, and a nitrogen base, 3 things. After we have that, we are then going to add another nucleotide to that. So DNA, if you guys remember, we went over in class, DNA is a double helix, which means it has 2 sides to it, or 2 strands of DNA to make up one DNA. As we add nucleotides, we're going to add them down to make up one of the strands. So what we're going to do is I'm going to add, coming off of the 3rd carbon here, we're going to add another phosphate. Coming off of that phosphate, so pretty much we're adding another nucleotide. Coming off of that phosphate, we're going to add another one of our 5-carbon sugars. All right, and then we get a different color here. We'll add a nitrogen base in the middle. We'll call this one G. Okay, so there is 2 nucleotides for DNA. Let me make one more, and then we'll be done with this side of our DNA. Another phosphate. I did real well there with keeping it to scale. Sorry, it's a little bit smaller than the other phosphates. It's still a phosphate. All right, and then lastly, let's put our nitrogen base in there. We'll call this one a T. All right, now the one thing I didn't go over yet is the bonds that are going to be between all of our characters in here. So we're going to have a bond connecting our 5-carbon sugar to our phosphate, connecting our 5-carbon sugar to our nitrogen base. Both of these here are going to be covalent bonds, all right? And then that's all we're going to worry about with bonds right now. And the other thing we got to go over to complete this first strand is the different sides of DNA. If you notice, this side has a phosphate pointing to the top, and this side here, pointing to the bottom, is the 5-carbon sugar. Well, we call them different things. If you look here, we go by the carbons, and the carbon, 3 is kind of pointing downward, and 5 is pointing upward. So the side with the phosphates pointing upward, we call this side 5', because the 5-carbon is pointing toward that side. And then the other one, the other side, this side here, if I label these carbons again, this would be 5, 4, 3, 2, and 1. This side here, we call 3'. All right, so because the 5th carbon is pointing upward, we call one side 5', And since the third carbon is pointing downward, we call that side 3'. All right. So now let me draw the other side of our DNA. Whatever this side is, because this is 5', this is 3', we could flip it. Whatever that side is, the other side is going to be the exact opposite of it. So in other words, we're going to flip that other side. This one's a little bit harder for me to draw, guys, so I'm going to do my best. So we're going to start off with our phosphate. Let's attach our 5-carbon sugar to it. All right. And then lastly, we're going to have our nitrogen base. We have our T there, so let's make a going to it. All right. And there's our first nucleotide of our other side of our DNA. And let me continue on here. We will make another nucleotide. And there's a G there. We'll make the C pink. Okay. So there's our C, and then one more nucleotide. And then there's an A. So let's attach our T to it. Okay. So now we have all of our DNA complete. We have our one strand on this side, other strand on this side. We have our phosphates in there, our 5-carbon sugars, our nitrogen bases in the middle. If you look at the outside of our DNA, in other words, if this was a ladder making up the outside of the ladder, we're going to have two things. So the phosphate and the 5-carbon sugar, that makes up what they call the backbone of our DNA. In other words, the outer parts of our DNA. So that's made up of two things, phosphate and our 5-carbon sugar. And then in the middle, in other words, the rungs of the ladder, those are made up of the nitrogen bases. We have A, T, G, C, apple in the tree, car in the garage. A always goes to T, G always goes to C, all right? Now, we haven't talked about how these are bonded together yet. So if we were to bond them, all right? Just make some little bonds in here, okay? These bonds right here in the middle, holding our nitrogen bases together, these are hydrogen bonds, okay? What these hydrogen bonds do, guys, is it keeps our two strands of DNA together. Now, we have covalent bonds holding our nucleotides together and holding the nucleotides to other nucleotides, but then we have the hydrogen bonds in the middle. The hydrogen bonds are much weaker than our covalent bonds. And since they're weaker, it's very easy for our DNA to just separate if it wants to, which we saw it separates during DNA replication. If you remember, helicase comes and breaks those hydrogen bonds in the middle. In other words, breaks these nucleotides apart right where the nitrogen bases bind to one another. Okay? So in other words, guys, if DNA is going to break, it's going to break in the middle. It's not going to break here. It's not going to break here. It breaks straight down the middle. All right? That's pretty much the structure of DNA. The only thing we have left here is on our other strand, do the primes. So if this is 3', this side over here automatically has to be 5'. If you don't remember that, just, carbons 1, 2, 3, 4, and 5. 5 is pointing that way. So that's 5'. That makes this side 3'. You can never have 5' and 5' right next to one another. It's always going to be 5 on one side and 3 on the other, okay? Other differences between DNA and RNA. DNA, just like this picture, is double-stranded. RNA is single-stranded. Okay. All of our nitrogen bases are the same for every single strand of DNA out there. We always have A, T, G, and C. For RNA, we still have our adenine, we still have our guanine, we still have our cytosine. The only difference is there is no thymine in RNA. Instead, there is a U. So in RNA, if we have DNA and it's A, RNA goes to U. So if we have DNA and it's an A, and RNA, A goes to U. Okay, but if DNA is a T, it's going to go to A for RNA. So still apple in the tree, car in the garage. If you guys want to do, like if you're doing like DNA to RNA, you can put the T's in originally for RNA, and then later on just edit it, take all the T's out, and put it in as U's. All right, so that's everything with DNA and RNA. Got a new page here. Next thing I wanted to go over with you guys is just protein synthesis. Okay, so I'm just going to give you a strand of our template DNA. All right, let's do G, A, G, C, A, T, G, 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 and then A. Okay. Now I know we talked about start codons and stop codons in class. I'm not going to worry too much about start and stop codons. Usually, guys, our DNA is going to be very, very, very long. Okay, DNA is going to be made up of millions of nucleotides all bound together, and that's just on one side of our double helix. So if we have this template strand of DNA, and again, guys, this is just a small section. Okay, our first thing is to get it to RNA. We term this transcription in class. We start off going to mRNA. And this process of going from DNA to RNA is called transcription, and it happens in our nucleus. All right, so we're going from DNA to mRNA. G goes to C, A goes to, well, it would be T, but there is no T in RNA, so it's going to go to U. G goes to C, C goes to G, A goes to U, T goes to A, G, 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 that would be straight C's there, and then A, it's going to be U, U, U. Okay? Now, when we are dealing with our mRNA, that's what we're going to use in those codon charts that I gave you in class. When you guys take your test, I know your test will be online for this stuff. The charts will be on the test. You'll have that as an attached image. So don't worry about having your own. I know a lot of yours are probably at school right now, so please don't worry about it. But we're going to use, I know a lot of you guys like to use the circle one better, so we'll use that. That's the one I put on your test. But we're always going to put our mRNA into that chart. Now, mRNA can then be transferred into tRNA, but I'm really not going to worry about the tRNA too much. I'll show you how to do mRNA to tRNA once, but the real goal of our tRNA or transfer RNA is simply to bring the amino acid in to bind to the mRNA. So it's pretty much going to be the same as our DNA, guys. C goes to G, U goes to A, C goes to G, all right? Now A would go to U, that's the difference, no T's, you know. Okay. And here's the goal of this tRNA. If we were to draw a picture of this, okay. All right. We're going to have a bunch of different nucleotides in here too, but the tRNA is going to have three main ones, and those three main ones are literally going to bind to the mRNA. And on the other side of this, let me draw the color here. On the other side of this, attached to the end of our tRNA, is going to be our amino acid. okay? And these amino acids, as they bind together, they make the protein, all right? So here's what we gotta do now. We gotta determine what amino acid is this in here that goes to the mRNA codon CUC, all right? So get your chart out, all right? C, start there, U, C, it's leucine, okay? So that's the amino acid that would be fused to our tRNA, and it's brought to bind to our mRNA. And I'm not going to draw that whole diagram for each one of these. Second one, GUA. Again, go to your chart, GUA. It's valine. Sorry guys, the noise in the background, the cats are playing. Next one, CCC, that's proline. And the last one, U is phenylalanine. I'm not going to write all phenylalanine out because you guys will use abbreviations anyway. But so I used CUC, I got leucine out of it. GUA, got valine out of it. CCC, proline, and UUU, phenylalanine. Okay, that's our amino acid sequence. Those amino acids will be bound together. That in the end will make up our protein. The process of going from our RNA to that amino acid, that is called translation, and it happens in the cytoplasm, specifically on our ribosome. Okay, the ribosomes could be the ones on the rough ER. That's if it's going to be exported out of the cell through our cell membrane, or they could also be the free ribosomes that are just floating throughout the cell. And if it's this amino acid sequence, if it's made on a free ribosome, that's when that protein is going to stay in the cell and be used in our cell. Okay, that's pretty much it, guys. All right, differences between DNA and RNA, there's your three. Deoxyribose is going to be in DNA. Okay, I'm telling you that'll be on a question on your test. Another one you could even do is DNA is longer and RNA is shorter. Reason being, guys, we're making a protein in the cell. There's going to be a bunch of proteins being made in your cells, like all the time, every day. If DNA is the instructions of your whole body, we're not going to need all of it to just make one single solitary protein. So we're not going to use all that DNA. So DNA is a lot longer. RNA is just going to have the instructions needed in order to make that specific protein. Okay. I hope this helped, guys. I hope the structure of DNA helped here. All right. If you have any questions, let me know. I'll talk to you guys later today.